Hello, everybody. My name is Catherine Bowers, and along with my co-chair, Tom Lurch, the organizing committee and the CAS organization, I welcome you to the ninth CAS Higher Order Structure Symposium. Now, it's been a challenging year, so I wish we could meet in person, but we do get to meet virtually, which I think is going to be a lot of fun. It's a new way of meeting, but you're going to meet some great people. Whether you're new to this conference or you've been with us all 9 years, I think you'll quickly learn that the people that attend this conference are not only great scientists, love proteins, but are just really, really nice people to work with. I think it's going to be a very interactive and engaging and a very scientifically rich conference. So, I really encourage you to ask questions, stay engaged, meet new people, come to the networking and the roundtables, the poster session. I think you all saw an email about the poster trivia, which is a lot of fun. Now, so get comfortable, grab your coffee or your tea, and comfortable the way you can only really be at home. Get your coffee and your tea and get ready for a great day of science. So with that, it is my honor to introduce our keynote speaker for today, Dr. Daria Mochly-Rosen. Dr. Mochly-Rosen comes to us from Stanford University School of Medicine, where she is the George D. Smith Professor of Translational Medicine and Professor in the Department of Chemical and Systems Biology. Dr. Mochly-Rosen is a protein chemist, which is awesome, who has a passion for molecular mechanisms that improve human health, focusing on both mitochondrial functions and cardiac and neurodegenerative disease. I think one of the things that I'm excited about most is hearing about her SPARK program, which is amazing in that it takes basic research and this wonderful research that occurs in universities across the world and helps to translate that into medicines to help human disease. So with that, I welcome you, Daria, and thank you so much for being a part of this conference.
Well, thank you, Catherine. And as you said, I'm a protein chemist by training. So yes, I do love proteins and I love being in your midst. So I will tell you about this program, SPARK, and hopefully I will get you excited to start programs like that in your own institution. So I'll cover three things. What is SPARK? Why do we do it? What are the goals and what do we achieve? And then I will spend a few minutes on kind of a simple guide from my own mistakes as an ex-entrepreneur in biotech. And then finally, I'll talk about SPARK Global. Why did we start it and why now? So let's start with SPARK, and the challenge that we are trying to address is that fantastic ideas from academia are basically dying on the vine, are published, but actually no one acts on that to make it into a solution for patients. And that is due to three gaps. The first gap is the industry doesn't view our project to be robust enough, and oftentimes are considered premature, either because the indication choice is not the right one, the solution is not realistic by their book, and also they find that our data are not reproducible. So that's the first gap, the value gap. And the second one is a knowledge gap. We in academia are absolutely clueless about what is industry doing and why our projects are considered premature or not robust enough by their criteria. We are not aware of what is drug development all about. And so as a result of that, we have a major disconnect with industry for that reason. And the third gap is cultural gap. We think of industry's work as kind of one foot in front of the other and not so creative, whereas industry, as you've heard, think of us as unreliable not realistic and so on. So to address these three gaps, and one more point, of course, there is an issue that if you are interested in translational research, that's not aligned with academic reward. That's not how you get professorship if you're just dealing with translational research. So the goal of Spark was to overcome these three gaps, and the solution were rather simple. So we wanted to make sure that good academic inventions do not die on the vine, and that they translate to benefit patients wherever they are. And that's why the first one is to increase the value of those academic inventions. And we are unique in that we are also focusing on academic inventions that are not necessarily providing economic outcomes that is particularly attractive. So we will choose also disease of developing countries or rare disease and the like. And we want to, of course, address the issue of gap of knowledge by teaching what it is that translation research is about. And we built unique educational programs that I will tell you about it in a bit. And finally, we need to address the fact that there is this mutual disrespect and address that directly. And the solution is particularly simple. All we do is bring the three cultures, the clinical and basic research culture, together with the industry culture into one room. By mixing people in the same room, naturally you overcome the disrespect. You spend time with a person, of course, you start to appreciate what they know and what you don't know. The way we do it is we have many industry advisors. They are all volunteers. They are all on confidentiality agreement. They join us now for 14 years in this now virtual room. Now, this is about half the rooms that we usually meet in. So every Wednesday night, 5:30 to 7:30, about 150 or 200 people are joining on our Zoom calls, or physically in our lecture hall when we can get back to it. And we used to do it on campus, and that's rather important. So it's not an incubator where you take an inventor outside, surround them with industry experts, they learn everything that needs to be done and move on, and then a new cadre of inventors are coming in. We do it on campus, so the know-how stays on in the university, and we also educate the educators, so they continue to teach it as new group of students come in. It is important that Spark is a place with no hierarchy. Perhaps it's easier in the US, but the meaning is that the meaning for no hierarchy, it means that a student in chemistry can advise a Nobel laureate in chemistry and vice versa, or a regulatory junior person can contradict a comment from an executive in big pharma. The reason for that is that it's only through floating many ideas that we make sure that the best idea is going forward. And if there were just one path to go forward in order to be successful, then industry would have been more successful. So it's really important this continues to have this atmosphere that is common in academia, which is out of the box thinking. We do not aim to reach a consensus. I am a strong believer that consensus is a killer of innovation. The consensus requires everybody to take the least risky path. And the least risky path is the one that was traveled before. And the one that was traveled before in the industry means that 9 out of 10 projects are going to fail. And that's when they start clinical studies. So if we are not keeping this culture, we are now in a first opportunity to try things out for the first time. And I have to say that one of the things that is clear to us is that the industry advisor, and usually we have about 40 of them in the room at any Wednesday meeting, they always had out-of-the-box ideas, but they could never float them because those were not accepted. Now they can live through us, and we try their crazy ideas and now either succeed or fail. And then after a few months, if it failed, we take another idea and continue with that. I want to say also that the out-of-the-box environments that we have allows really to address, to look at problems differently than it has been looked at before. And that is part of the reasons why the industry advisor, when they walk out of the room every Wednesday night, they say, wow, that was fun. It was fun because it allowed them to float ideas, to hear other people comment on them, and to see the execution that occurs as a result of the discussion. And it is important to learn from failure more than to learn from success. We all know that from engineering, but somehow in pharma, it's not something that we see happening. Industry is not interested in educate other companies what not to do. And also they don't have the time to write about failures and need to move to the next project. So this is an environment where industry advisors can share stories about failures without disclosing their own company's secret, but enough so that we can learn from it. So the formula for success is doing it in one room and ongoing with many advisors, not trying to reach a consensus and trying ideas in real time. So every week we have either a lecture or two from our industry advisors, and in alternate weeks we have project updates from 4 projects that are in the program. And the structure for the project update is very strict, where most of the time you spend on, here are my problems, and that's the issues that I would like to have advice on, rather than look at me, I've achieved this and the other things. So it's very different from our usual conference of sharing science. So Spark has been in operation now for 14 years. We are on the 15th year, and our success rate remains around 60%. And that success rate is very high. Success measured as projects that moved from an idea or a patent even disclosure or paper to industry's hand and or to clinical study. That success rate is about 6 times higher than the best success rate that industry reports about themselves or VC report about their programs. We also found that there are other aspects that are achieved as a result of Spark, which perhaps will be matter more for your institution, which is that we provide very little money for each project, about $50,000 a project a year for 2 years. Whereas we find that they are Sparkee, the people that have been participants in the program, achieve about $8 in grants for each $1 that they got from Spark. So the academic institution is, of course, very happy with that, as it brings indirect cost to the institution. We also continue to do our academic mission, which is publish. And in fact, we get reports from our Sparkees that they publish at least 3 papers based on the works that they've done in Spark. They are also very successful in raising money for their startup. And in last studies that we've done, there were $16 million raised for each startup that came from Spark. And the attrition rate, in other words, companies that fold, is very low. Only 10% of the projects that started as a company fold after a while. And I'll come back to this point in a moment. So here are some of the startup and the pharma partners that resulted as a result of our activities. And this is incomplete list, but I want to focus on one case since it fits best for Cass, and not talking about my own research, this will be my favorite one, which is a disease called a hereditary transthyretin ameliodosis, which is a very rare disease. There are only 50,000 people around the world. It's a genetic disease due to many mutations in this protein. And as a result of improper folding of the protein, amyloids are being formed. It occurs in adults. It causes heart failure, kidney failure, and so on. And so patients are born with this mutation, but the disease actually manifests itself later in life. And the reason for that is simple. It's a tetrameric protein that, because of the mutation, tetramer formation is not stable. The tetramer form is not stable enough, and then the monomer starts to misfold and create the amyloid polymers. So Isabella Graff was an assistant professor in the Department of Pathology. She came to us with this project because she found a small molecule that seems to stabilize a tetramer. This small molecule was AG10 in her named by her. And importantly, she was frustrated because she wrote patents about it, she published about it, and industry was not interested. And so she came to Spark. And we helped her not only with chemistry advice and formulation, but also with introduction to companies, with packaging her story in more effective way. And the bottom line is that she built Ideos, and Ideos quickly went public, and they raised a lot of money from the first round, and eventually was acquired by BridgeBio, and now they are completing phase 3, and based on phase 2, I'm very optimistic that indeed they will have a drug on the market. So this is an example of a project that started from an early observation by somebody who has no chemistry background, but actually much more a biologist that got all the way through hopefully phase 3 now. So I mentioned that our attrition rate is 10%, and that's 4 out of the 43 companies from the last questionnaires that we sent out. And that is particularly low. There was a paper that was published in Nature Biotechnology in February of last year that examined all the startups that were university-based startup and what their success rate. And they reported that about 70% of those are economically invaluable. Some of them, many of them have folded, and others are just teetering and about to fail. So a 10% attrition rate versus 70% or 80% attrition rate is a big difference. And so why is attrition rate so low? It's not that our invention and inventor are so good or particularly better than other inventions that go on. It's really because they are better partners for their pharma or VCs. And we heard it from Isabella, and we hear it from many others, where they hear again and again that they are surprised how mature the project is, and more importantly, how well informed is the inventor. They also report to us back that the projects have been already vetted by industry experts, and so by that they are much more of interest to industry. And finally, our inventors tell us that by being in Spark and being exposed to so many veterans and current employee of Big Pharma, they have many connections and can really float their project faster, better into the hands of the next developers, the startup or the investor or the pharma partner. So what do we actually teach? I think that the most difficult things that we have to teach is how to think about your project from the end to where you are and what are the gaps. So in our case, in Spark, our ends are drug development. There are other Sparks that are aiming, for example, to develop a device. But the point is very similar. If you know where you're going, who is your patient, who is your physician, what the family like, what is the condition to give the drug, and where you are, you can identify quickly gaps in the way you're thinking about it, and then quickly focus on closing that gaps rather than continuing with a typical academic kind of random walk. It's important to be very intentional when you're doing drug development. That's different from our basic research. So because of that, we immediately teach that aspect as well by using money. So money is given to the project based on milestones. Now, remember that I mentioned $50,000 a project a year. Academic labs at Stanford have between $500,000 to $1 million a year. So 50,000 is not really a huge incentive. And importantly, it doesn't go to their account right away. And yet, the idea that your project is being funded only through a milestone, and then the rest of the money will be given to you only if you reached it, or will go to another project, is a very important educational tool that stays with the inventor should they go to industry. That's how projects are funded in industry. We teach project management skill. It's another thing that we are oftentimes clueless about in academia, which is making sure that all the process that needs to be completed before you move to this next step are actually in alignment, and that you are not creating time gap, because time gap is money, and therefore increasing the chance that you will not match and meet your milestone and fail. We also very much aware that we in academia are not aware of what drug development is, and so we provide this education. And through that, we prepare our workforce, students, postdocs that may go to industry, and many of them will do that. We teach them how to do that. We don't count on my 1 year in industry to teach this. We actually, as I said earlier, have many industry veterans. We started with 5. We now have over 150 people who are willing and volunteered to provide advice in this program. And in fact, at times we have to turn down people because when we met in a physical space, we just couldn't have so many people in one room. And we, of course, through these advisors, we provide introduction to not only collaborators, even within our scientists at Stanford, but also with companies, contractors, services, and investors. And we do teach entrepreneurship, although I have to say that we were doing it initially quite reluctantly because we felt that our academic work should stop before that. But we now know that entrepreneurship is something that is very coveted by our students and postdocs. Okay. So since we teach entrepreneurship, I'm going to give you a brief guide based on my own experience as entrepreneur. And I have to say that most of my advice, which are summarized in one slide, came from mistakes I've made. I think that we learn much more from mistakes than from successes. And in fact, every Spark meeting starts with me giving 5 minutes lesson learned from mistakes. And most of them I give because somehow people are embarrassed to share their mistakes or mistakes of their colleagues. I hope that our 5 minutes mistake program will be eventually available for others to view. I think that it's a useful tool. All right, so here's me quite a few years ago, 2004, September. I'm sitting in Chicago Airport Hotel. I'm sitting down because, and around me are members of the companies that I founded with my graduate students, Leon Chen, called Kai Pharmaceuticals. Next to me on the left is Kevin Grimes, that is co-director of Spark at Stanford. He was then the CMO of the company. And I'm sitting down because I'm activating the first site that is going to use a peptide that is protein-protein interaction inhibitors for patients with acute myocardial infarction, with heart attack, delivered into their coronary during the procedures that open up the occlusion to the blood vessel. The reason why I'm at the hotel is that inside the lecture hall there are 70 representatives of clinical sites that are going to use this drug and their nurses, so about 150 people in the room. I just finished telling them about the project, and I got a page that the first patient has consented to participate in a clinical trial in Belmont, I believe. And I'm sitting down because all I had to do is push the return button on the computer, but I could not stand up. The realization now that this peptide that we developed at the bench at Stanford for just to prove a working hypothesis about the importance of protein-protein interaction, not with goals to develop drugs, turn out to be a drug that now a patient and their physician and their family consented to be used. So really an overwhelming moment. I was in the company for a year, helping set it up. Then I moved back to Stanford, and when I came back to Stanford, I realized that year that I spent in the company taught me more than I learned in the many years prior to that. And so I suggested to the dean to start this Spark program. And after a year, I realized that Kevin, an MD, MBA, will be a perfect partner to my PhD protein chemistry. And that's how I actually, just showing you my priority, I recruited him from the company to come back to Stanford, and he's now a professor in our department and my partner in crime. So this heart attack drug was not eventually developing fully to phase 3 because of actually business decision. And in fact, the products that the company eventually was acquired by Amgen is a product that called Parsabiv for patients with that are on kidney dialysis. And the reason why this drug was developed by Kai is actually one of the lessons that I will mention in a second. Okay, so in a nutshell, 10 lessons about entrepreneurship. The first one is how important it is to talk to as many people as you can about your project, your idea, the clinical indication, how to build a business plan. Recognize that there is nothing in your career until this point that really prepared you to be an entrepreneur. So you have to consult, and you have to consult as much as you can and as effectively as you can. One of the things that really matters in consulting is to be very clear about what you want from the person you speak with, and spend as little time explaining yourself and talking about your idea, and as much time listening to advice. So that's when another challenge comes about, which is to prepare your idea in what is called elevator pitch, in a very short, succinct way to express the idea so that you can spend most of the time hearing rather than speaking. Not natural for academician. We are used to, the moment we get the podium, to speak at no end. All right, the second one is dare. You're an academician, you're a professor in your university for a while. It is risky to start to do translational research. You have to realize that every step of the way, the chances for you to fail is greater than the chances to succeed. And I think that there is a lot of expectation, especially initially, that you will be successful since you've reached the accolades that you have. Surely you can do that. And if that's the expectation, you're going to fail, and it will be difficult to get up. So basically, every event should be an independent event, and it doesn't matter that you went and pitched your idea 100 times, because if the 101 is the one that find you the partner to continue to work with, who cares about the 100 no's that you've heard? And I definitely heard at least 100 no's before I got the first yes. And that's when perseverance is really important. So if you go into this meeting feeling every time hurt because they didn't appreciate how smart you are or how good the idea is, or and so on and so forth, then you won't be able to persevere. But if it's about your project and why the patient matters to you, and you have that in mind, then it doesn't matter. Another lesson that oftentimes you get conflicting information about, and I hope that you will accept mine, is to diversify. What do I mean by that? We oftentimes have a single idea coming from our lab that is a basis for our interest in translating it to product. But a single idea, the chances it will succeed is very low. In fact, in industry, when a project enters clinical trial, only 1 in 10 eventually reaches approval. So you have 2 options to diversify. The first one is to be your own devil's advocate, think what's wrong about your project and build alternative to your solution. While you're building your first solution, builds 3, 4 other solutions to the same problem. So let's say siRNA to reduce the level of the protein that is misfolded, peptides that inhibits the misfolding, small molecules that increase the proper folding, as an example. So you have now 3 shots at the goal. You're developing them in parallel. The chances of success of one of these goals is higher. The second one is to think about colleagues in your own campus or elsewhere, let's say those that you meet in the CAS meeting, that are working on a similar problem but have different solutions, and talk to them into joining forces for one adventure. So that means that you're going to divide the pie, and it's rather important that you decide on the division of the pie before you know which one is successful, recognizing that, let's say you have 3 projects, you should have 1/3 ownership in each project. The advantage of that approach is that you have more chances to succeed. So if we are talking about 1 in 10, if you have 3 projects, you have much better chance than if you have 1 project. If you do something like that, where you combine multiple inventions, you have to work with your university to make sure that all the universities are on the same page. And there is a lot of advice on that as well, not here. And also that you are working with people that you have mutual respect for, because there is nothing that kills a startup as animosity between the inventors. So that's another thing that's important, that when you are in conference like this one, you spend time actually become friends of your competitors and work with them as colleagues rather than competitors. When you start a company, the first thing that happens is that the moment you enter clinical studies, there is tendency by the VC to kill basic research in the company because it's money, and money is limited. It is important that you keep basic research. And as an entrepreneur for the first time, the one thing that can really help you is to make sure that as you sit on the board in the beginning, when everybody is still in the honeymoon, you make sure that in your resolutions of the board, there is a decision of how much money will go to basic research, no matter what. And luckily for me, I've done that based on advice of another entrepreneur whose company folded very early, and ensure that 10% of all money always goes to basic research. And that's part of the reason why we developed Parsabiv. We had the side effect from a drug that was in clinical studies that was unexpected, that caused a fast lowering of calcium in older patients. And that side effects was catastrophic. We couldn't continue beyond phase 1. But because we had first of all, we had a very good CMO that helped us think through that. And because we had resources in the basic research, we spent time thinking, is there an indication where lowering calcium will be advantageous? And that's how we came to think about patients on kidney dialysis. And the story is a bit longer, but this is a story of making a lemonade from a lemon. That tells you why it's important that when a project fails or appears to fail, that you have to spend time thinking before you give up. I have to say that the VCs, the moment they heard the side effects, they said, okay, we close the company. But we did have some cash left in the company, and we felt that we can convince them to continue the study based on some of the basic research that was done very quickly in the first couple of weeks after we found out of this failure. So by viewing every event as an opportunity rather than first as a failure, you can really come stronger from such an event. And I have to say that I had very little respect for the business development group initially. Don't tell them that. But I have to say that they were instrumental in our success, because when we started to develop Parsabiv, we knew that there is a drug on the market already, sold by Amgen. It's orally available. Ours was IV. And we thought, and it's very cheap, and it's about to become generic. So we were kind of worried about what can we really do that will be used. Why would our invention be the one that will take the market? And the reason was that our business people actually found that there are problems with the current drug. It causes nausea and vomiting. Patients don't like it and they don't use it. These are patients that come every week, 3 times a week for dialysis. Their vein is open because they do dialysis. So actually giving a drug through the IV every 2 days or so is very acceptable for this patient population. So we knew that we have to make sure that our drug doesn't cause the stomach trouble. And since it didn't, that drug has been taken the market, and actually Amgen is the one that acquired the company. And finally, this is something that we all know. We all say the strength of the chain is the strength of the weakest link. we say it, but we don't mean it. In a company, there is nothing that is truer, and I can give you multiple examples of how important it was to make sure that every member in the of the company mattered. And one simple way that we've done it is to every week, every Friday, meet for an hour with everybody in the company about what was done this week, what are the challenges for next week, what are we worried about, what are we happy about. Everyone from the assistant to the IT person to the rat cage washer to the CEO were sitting in the room. And that was really important. It was always a kind of a happy hour, but really critical for the reasons why eventually the company was successful. So I told you quite a lot about my experience as an entrepreneur, and I want to finish with telling you a little bit about why Spark and why Spark Global. So we built the program initially for Stanford because we believe that it is really our social responsibility to take early discoveries and make sure that they move to industry's hand to benefit society. And the source for that belief is because we are funded by society, by public. We are taxpayer funded academia. And so recognizing that it is our responsibility to make sure that our ideas are developed is important. It does not mean that individuals in each academic institution are responsible for that, but it is the institution that needs to have this program. So I hope that clear to you, and that the institution will build program of translation research. It's also important for academic institutions to do that because, as I mentioned earlier, most of our students and postdocs are more likely to hold positions in industry rather than in academia, because we professors in academia, we don't die and we do not retire. And so the number of positions available for them is rather limited. So we need to teach them, to prepare them for the jobs that they will hold. And I can tell you that at Stanford, I've taken the best students and postdocs to Kai, and together with them, I learned how clueless we were, all of us, in what translation research is about, how not intuitive it is, and how intellectually challenging. And the other reasons to build a SPARK program is that there is a potential economic outcome for the area where the university is. So at Stanford, to build a Spark perhaps is less important because we have biotech and investors and so on in the Silicon Valley everywhere. But if you are moving out of this belt of areas where there is more biotech to areas that are have fewer of them, if you develop a program like Spark, the chances are that the companies will be formed locally and therefore will improve the economic outcome of the area around the university. This is an incentive not only for the university, but also for the government, the local government. And that is another tool that you could use to convince them to invest in Spark in your institution. So how do you begin with that? If you wish to start Spark in your own universities, you have to have internal champion. I can tell you that people have assumed again and again that at Stanford, it's no problem, that everybody is entrepreneur and we don't need much in the way of champion support of that. But you are actually wrong. Even a Nobel laureate that has been instrumental in starting biotech industry per se was very much against translation research. When I put ads for the program, he brought one of the ads to the dean's office, where I was then a senior associate dean, and said, "What are you doing? This should not be work that academia should be busy with. It is only for profit, and we are doing research for the sake of knowledge," and on and on. So there was a lot of resistance to that. And it was also in a year in 2007 or so, 6, 7, where there was very limited money for basic research. And so there was feelings that if we have money to support translation research, it should be given to basic research. But one way that I persevered is that one of the most loud objector to the program had a patent that was not licensed. So I invited him to take his patent and come to the program and try to license it through learning more what's wrong with his patent. And luckily for me, he joined. Unluckily for him, he was one of actually the only project from that year that failed. But as a result of going through Spark, he became the strongest advocate of the program and has changed his career trajectory and has been very, very strong supporter of the program in the initial years. You need to find directors that have some relevant experience. So my 1 year in industry doesn't qualify me, but it was important that I was also a professor at Stanford. So that when I talked to other professors about translation research, they knew that I shared their experience about worrying about grants and education of their students and so on, not only coming from industry with the experience of running a large company. So it's important that you have one director with industry experience and another director that is academics, if you can. It is important to count on industry volunteers, not to pay. If you pay one advisor, you have to pay them all, and you cannot afford it because each hour is what, $500 to maybe $5,000, depending on the advisor. So it's important that there will be volunteers. And if you make a fun program where they feel that they are heard, they will come. And they will come even in areas where there is no pharma. So oftentimes, people who start Spark outside the main pharma belt worry about how do they find volunteers in such an area. And the answer is actually easy. If you were an academic institution and you taught people sciences, they have gone to work in companies, they may retire and come back to the areas to be with their grandchildren, they are there. So start with alumni and build first the program through the alumni, and eventually you enrich it through people who are willing to commute. We, for example, have an advisor that is in Washington State that comes every Wednesday night, used to come every Wednesday night to our meetings, flying on his own cost without being reimbursed for that. And the reason for that is that forum of advisors is fun for them as well. It's a kind of a social club. They also enjoy the fact that they are so impactful on our projects. They learn the latest science. So you can get many advisors to contribute to your program. And of course, now with Zoom, since we got used to that, perhaps you can do it also via Zoom. Also, I have to say that informal interactions will not replace Zoom. And I'm sure that you'll see it also with this conference. All these side chats are critical. For your university to have Spark, you have to have core facilities that are helping translation research. You don't have to have all of them, but it's good that you have some. For example, animal behavior facility or high throughput screen facility or proteomics and so on. And if you don't have them, through the network, you can find out other universities that have it and make sure that you have access to it. You need some funding, especially for the project managers. So the university have to pay FTEs, but you need some funding also for the project, because especially in the beginning, the funding that the academic have for the project is not a translational aspect. And since it takes them time to raise money based on translation research, you need to help them initially. And of course, as always, you need to be very flexible. So we started Spark in 2005 at Stanford, 2005, 2006. And a few years later, the government of Taiwan has decided that they are interested in developing biotech on the island. And they reached out to me to help them start a program like that in Taiwan. They started with 7 universities that had Spark program. They fund them actually 4 times higher per project than we do. But what they've done, which we have not done, was to write about it. So once they wrote about Spark in Taiwan, other countries have reached us, and we're now facing about 60 Sparks throughout the world. some of the program are very advanced, like the one in Germany, the one in Finland, the one in Israel, and others are more just beginning, including in the US. So Spark Global Value is not different from Spark at Stanford Value. We are a non-for-profit organization working within academia. It's important that it will be ethical for patients and really using the highest industry standard, that we are building on volunteer spirit, so not paying the advisors, but making sure that the advisors see the benefit of participating in that stage of drug development, the early stage, so that we increase the pipeline for them eventually, not necessarily individually, but as an industry. And the value that is really running it is that it is for patients and society. And one of the things that we've discovered was how much waste of terrific ideas occur, especially if you are not necessarily in the better known universities or the countries that are better known for sciences. We've seen again and again amazing science done everywhere. And I can tell you, for example, we started Spark program in Zimbabwe, and I remember somebody telling me, well, Zimbabwe needs other things. They don't need biotech companies. First of all, who are we to say what a country needs or doesn't need? It's the decisions that locals should make, but also the kind of science that Spark Zimbabwe is doing, which is pharmacogenomics, is really breakthrough, fantastic and high quality research. And more importantly, it's impactful not only for them, but also for us. They developed a program that we are now helping implement here based on their know-how. What a waste it is if good ideas don't come to fruition. And so what we are trying to do with Spark Global is to make sure that we are less wasteful with good ideas, because there are so many problems that are remaining unsolved. So I hope that I convince you that Spark Global matters, but I also have another goal for our global organization, and that is that when we are having scientists throughout the world that are more familiar with what translational research is about, we have an opportunity to recruit them to address new threats. And of course, this threat was very much on our mind in the past past year. So in April, or actually towards the beginning of April, when the extent of the pandemic became clear, I wrote to all the Spark directors that have started program or have active program and asked them if their institution has active research that relates to COVID. And if they do they need help with advisors? We have 150 advisors or more. They usually have fewer. And so basically volunteering our volunteers to help with these projects. And I got quite a few responses. In the first selection, there were over 50 projects that people were working on throughout the world. I have to say that there are many more projects than that after a year. But my point was that there was an opportunity to combine forces between Sparks in various nodes to address it. And it addresses, for example, just sequencing and contributing to the database of mutations in the virus that evolved. This is Spark in Kuala Nazaru in South Africa. They have fantastic contribution there. For diagnostics, there are multiple groups that have worked on diagnostics and have, some of them have progressed hopefully to the market, but definitely just sequencing. So many people were just doing sequencing as a way to make sure that we know patients that are carrying the virus and those that are just having a cold. And finally, for therapeutics, there were quite a few projects, including a project that eventually connected Stanford to Spark Australia that had to do with prophylaxis, that we now progressed all the way to completing phase 1 and hopefully will continue to move forward. So just to summarize what I told you, told you about my own experience in entrepreneur and how I was not prepared to starting a company, even though I used to teach drug discovery and development, still do at Stanford. But I taught it before I started it. And now I realize that it was based on what I learned from a book. And it's very far from what you learn when you actually go through that. Spark provides an opportunity for us to pay back our debt to society for allowing us to do exploratory science that is so rewarding. I've spoke a little bit about my own experience as an entrepreneur and mistakes that I've made. Unfortunately, I didn't have time to tell you long stories, but those perhaps will be another time. Where I thought that it's intuitive and I can figure out my way. And as a result of that, made many mistakes. I'll give you one example that you can appreciate. When I mentioned that we have a peptide that was used for acute myocardial infarction and was given into a coronary artery to the site of infarction using the balloon catheter that is opening the occlusion. When we did this study at Stanford, we've started it initially, we've done it in rats, and then I gave it to competitors and collaborators, and they've tried it in a number of other species. And eventually, I was told that I need to do it in large animals. And at Stanford, there was access to pigs. A lot of the cardiologists were developing devices for coronary artery occlusion, and so they were using pigs for that model. And so I decided I will try our peptide in a pig. But as I was starting these experiments, I wanted to make sure that what we provide as a treatment, the peptide, will be very pure. So I spent a lot of money and time to get to a 99.5% pure peptide before I used it in the animal. And then we've done the study in the animal, and the data were fantastic. A 70% reduction in infarct size in animals that had an infarct that should, in human, would cause heart failure and death. And then we started to, we got investors and we started to move forward with the development of the drug, and we hired an executive for drug development. And I told her that we use 99% pure peptide. And she said, "This is awful." And I said, "Well, we couldn't make it 100% pure no matter what we tried." And she said, "No, this is awful. You cannot use a drug that is 99% pure because you cannot scale it up. You have to use a drug that you eventually can produce in large amounts. And if it's so pure, you will never be able to produce drug in high amounts. And if you've done your studies with such a pure drug, you have to repeat it with less pure drug." And so we had to repeat our studies with 95% pure peptide rather than 99% pure. And I thought I did the right thing. I just wasted money, time, and it was only because I never asked anyone. I thought I could figure it out. So I want to use this simple story as a reminder of how unintuitive drug development is and how you need to count on people's advice. And finally, I hope that I convince you that Spark is worthwhile activities in your own institution. And I really hope that you will join in Spark Global, because we can really make a difference in the cost of drug development, in making sure that we are benefiting society, and also that we are addressing need wherever they arise based on our capabilities of solving problems, not because we care about making money. So the guides for Spark is published. It's not really a full description of all the things that you need to do with in order to translate research. And the reason why I'm comfortable showing you the book is you can buy it on Amazon, but we did not get $1 for writing it or publishing or selling it, because we thought that the company Springer will sell it for really cheaply. Unfortunately, it's not that cheap. But at any rate, it provides a way for you to start to realize what it is that you need to know for drug discovery and development. And the book is full of anecdotes in the gray boxes of mistakes that I made. It's called What Surprised an Academician. And so it is a means to remind us again and again that it is not intuitive and smart is not enough. I'm sure other entrepreneurs can fill up the rest of the gray boxes. So I look forward to hearing from you. And I am so grateful to have the opportunity to open this conference. Protein is my love. And I hope that you are using your love to make sure that we improve patients. So thank you for your attention.
Oh my goodness. Thank you, Daria, for a wonderful presentation. I have such a genuine respect for your passion for not only science, but just collaboration across all the different aspects of drug development. As I'm listening to you, I want to say, choose me. Let me be a part of it. I mean, that's how excited I am after listening to your talk. I just cannot tell you how wonderful your approach is to me. It really speaks to my heart. This was a wonderful way for the CAS HOS meeting to open, where we strive to be a community that does bring students and industry and regulators together. So I hope everyone that's in this meeting today has been motivated by your talk. It was wonderful. It was wonderful. So with that, we do have a number of questions, and if you're okay, I'll start asking away.
Please do.
Okay. All right. The first question, how do you fit patient convenience in your drug design? Is it at all a consideration, or is speed to clinic market a much more influencing factor?
Excellent question. And when somebody sounds like quite savvy. So I mentioned from the beginning that you have to think about your project from the end with the end in mind. So this is exactly what you need to consider: patient convenience, cost, physicians, their surrounding, what temperature is your drug going to be traveling from one place to the other? Is it going to use, a cold chain transfer, or is it going to be fine to send by in an envelope, and so on and so forth? So we do consider it, and we do emphasize it when we are in the planning phase. However, the goal of Spark is not the drug company goal. We are just trying to get into proof of concept sufficiently advanced so that investors or companies see the value of it. And sometimes proof of concept may be the less convenient solution just to show that it works in human. So for example, if you are thinking about convenience for patients to have an oral drug, but at the moment you have only injectable drug, and you have a good biomarker for the disease that will show that indeed, or clinical endpoint that will show that the disease can be addressed by their compound, and you only have injectable material this time because you didn't figure out the formulation, go with the injectable and do a small phase 2A after the safety study with an injectable. But you need to be aware of that this is not the optimal solution for the final product. And it will be very savvy of you when you are presenting your idea saying, this is how far I got. The same goes with animal studies. It's easier to inject animals intraperitoneally than IV, or to formulate it so they will take it orally in a reliable fashion. So it is okay to start with IP injection, but you need to say when you are going and pitching your idea, this is the first thing we've done, but it's actually water soluble, and it's not, it can be absorbed through the stomach, because when we do gavage, we can show that it gets the same effect, but we could not formulate it for these studies, and therefore we've done our animal studies with intraperitoneal injection. In other words, being aware of it is 3/4 of the way to convince others that it is okay to go with your perhaps less patient comfortable solution.
Awesome. Next question. Any thoughts on how to change the incentive system that leads to the publication of results that are not reproducible or robust?
Okay. This is a lecture on its own, and we had Glenn Begley at Spark every year. He's Australian, the Australian that actually shook our academic confidence by showing that our data are not reproducible when he was at Amgen. There were other papers that showed the same thing, and basically they said that our the best publication oftentimes are not robust enough for a variety of reasons. We are rushing to publication. We are in love with our idea and kind of having blind sight to any things that didn't work, and so on and so forth. And so the incentive to publish, is stronger than the incentive to publish right. Lots of partners at fault. The journals, the reward system in the university, the ego that we have, lots of problems. But I want to just talk about as one simple solution. So those problems are for the institution to address. And for example, at Stanford, we have now a program that focuses on that alone. I want to point out one thing. If the investigator is truly rooting for their idea to be a solution for patients, they should be open enough to this criticism. And so if a company reach out to them and say, well, I tried what you were doing and it's not working, work with them. Have them come to your lab. You go to their lab or send a researcher to their labs to make sure that you really figure out what does not work. If you are open to that, you are already half of the way through that so that people will not waste their time and money. It is not like a great recipe where you are missing an important ingredient because you don't want anybody else to cook as well as you do. It's really important to be able to reproduce your work. So simple tricks that I use in my lab is before we publish, I have somebody take the methods that were provided one person and repeat the experiment independently. I have to say that it was initially difficult to implement, not foolproof anyhow, but the only incentives that a student has when I ask them to repeat somebody else's work is that eventually somebody else will repeat their work as well. So if you can manage to do that, I recommend it. I have to say that, we all have flaws in that, and it's not foolproof, but it's another method for that. For the institution, stop blaming the faculty. If something doesn't work and immediately there is a blame or misconduct and so on, assume first just innocent sloppiness or innocent mistake rather than that. And one more point, we have gone through this now many times where actually lack of reproducibility has to do with difference between the way the experiment is done, that people are adopting changes that they think are not making any difference, and as a result of that, the experiment does not work. And we have quite a few stories with Sparkees that work with big pharma that took their technology, and only because they kept coming to the companies, they realized that they are doing things differently. And one good story is one that is for healing of a perforated eardrum, in which the company didn't know anything about the ear structure and basically put the drug in the wrong place in the ear. And so no wonder it didn't work. So sometimes we need to work on to work as good partners for the company and not immediately revert to this, it's my fault, it's your fault. Work together to make sure that the solution is really tested as closely as it was tested in your hands, and see if it was a fluke or it's real.
Very cool. Okay, next question. This attendee says, hi, Daria. Greeting and thanks for the presentation. I wanted to know what is the next step towards starting a Spark program in a university once the 6 points mentioned in how to begin slide have been figured out. Does the university then reach out to the Spark global team?
Okay, so the first thing, if you want to start a Spark program in your own institution, email me and we will meet with you and talk you through what needs to be done, understand whether you are working on your own or actually your university dean or director or whatever is interested in the program. And we have a starting package to help you figure out how to work your way. We help you recruit advisors and so on. So yeah, the Spark, now we have an executive director for the global program because we spend a lot of time helping others. And so, yeah, we are happy to help you. And so you start with an email to me and I'll be delighted to help you get started.
Awesome. And if anybody starts a Spark program in North Carolina, let me know.
We don't have Spark in North Carolina.
No. So let's do it. A final question. How does the team address academic science that is not yet ready for progress from idea to company? For example, where a professor is a strong champion for his or her idea, but is not receptive to pitfalls.
Okay, so what you're saying is that how does a program deals with the problematic inventors and Mr. Know-it-all that is not listening, right?
I think that might be the general question.
Okay, so I just want to make sure that I translate it to something I understand. It happens, and it's rather an interesting process. Rarely we just disengage with them. So I think in our 14 years, we had, I think maybe 2 cases where we said, okay, you're done. This is not working. It's actually rather interesting because we mix projects and nobody really asked about that, but we take projects that are really an idea. I have a phenotype in my mouse that is unexpected as a result of knocking down a gene, and I want to explore how to target that gene product. Or I use a drug in patients and suddenly they have this unexpected effect, and I want to explore the mechanism of that and anything in between. So project can be really far in the clinic or can be really early or anything in between. And we have projects not only by faculty, but we have few projects by students because those are the students that went through 2 quarters of drug discovery and development. And they have teams of students that are working on projects that deemed worthy by advisors to move forward. So when we have project updates, and I said it starts with actually the first meeting of every year, which is January, start with what is called target product profile. It's another good story when I was starting the company and the same VC for drug development asked me, what is a TPP? What is my TPP? And then I had to understand what she says, target product profile. And then I said, oh, come on. We'll know it when we see it. Why waste time and sit here and describe? Of course, it is essential. So we start with target product profile. And oftentimes we'll put one of the student teams first, and you'll see the professors. Now I don't see it anymore, but when we were in one room, I would see them working on their slides and fixing their target product profile as they listen to somebody else's. So there is a lot of lateral learning by examples. And also every year, since the project is in the program for 2 years, they also hear advanced programs. So they see, for example, I don't know, Stan Cohen, whose invention started the recombinant, it's a Boyer-Cohen patent, started the world of biotech. He's a Sparky, and they see him listening to advice and confining to the slide structure and so on. So they come to shape. but it's not working 100%. We have an inventor that had a really fantastic program that showed really great efficacy in animals, and we were pushing them to go forward with finding a partner for it. And instead, they start to do fancy schmancy stuff, put stem cell in it, and all sorts of things that are impractical and nice research, but are not a priority if you're really interested in solving a clinical problem for patients. So,
Daria, so we are now out of time. I'm
yeah.
sad. I would love to keep talking, but thank you so much for joining us and being a wonderful part of opening this conference for us. I encourage everybody to reach out to Daria if you have any further questions or interested in Spark. Thank you so much.
Thank you for the opportunity, and have fun in the meeting.
Thank you.